that you have to know it or memorize it. Let's start with it. Nickname is Ala. That is from alanine, so we can say Ala, the initial A, H3, which is a methyl group. Okay, so that is make the alanine, it's like a chiral, not a chiral. All the amino acids are chiral except the glycine. Remember this information. So the alanine is an important in the glucose alanine cycle. How is that? Because the glucose converted to the pyruvate, and then the pyruvate by the ALT converted to the alanine by the help of a glutamate, so that is well transferred to the alpha ketoglutarate acid, and we can see that again. It's contributed to the structure of protein, of course, and alanine has two versions, D form and L form. That's why the glycine doesn't have this form, because it's achiral. The glycine is achiral because there is no D or L form. There is nothing is called the mirror image, but the alanine has two versions, D form and L form. More common is L form. D form is just found, you know, that D is mean dextro, that's mean it rotate to the right, and L form, that's mean rotate to the left. D-alanine used by bacteria, so it can be used in the antibiotic. No, I mean the D-alanine is in the formation of the wall of the bacteria. So we can use this information for using of antibiotics because they attack the D-alanine. Side chain is methyl group, which is there. It's a methyl group, which is known as a methyl group. So remember, the side chain of alanine is the methyl group. The second smallest flexible generic. Okay, it's the second smallest amino acid. What is the smallest amino acid? Which is glycine. So the glycine is the first amino acid, the smallest, I mean, because the side group is H. And here the second smallest is alanine because it contains the methyl group, which is known as a CH3. Again, all amino acids except the glycine are chiral. All the amino acids are chiral. That means they have a mirror image, they have D, they have L, they can rotate, but the glycine cannot rotate, you know, because it contains H group, so it can be as a mirror image. Okay, if there is a chiral center, it's stereoisomer. So the glycine, it's achiral. It means if it's stereoisomer, it can be more opposite direction. Okay, D, that's mean rotate to the right. L, that's mean rotate to the left. The bacterial cell wall formed of the alanine inside the peptidoglycan, because we know that the wall of the bacteria is formed from peptidoglycan. Inside the peptidoglycan there is something is called the D-alanine. The bacteria use the form of alanine in their cell wall in the help of a transpeptase. Remember, the D-alanine it's not common at all, and it's present in the wall of the bacteria. Ampicillin and antibiotics, which is attack the cell wall, attacks especially the D-alanine. Remember also that the alanine is non-polar, non-charged, because its side group is a CH3, so there is nothing make it polar, so it's not polar, it's not a charge, and it's hydrophobic. Non-essential, that's mean because we can make it from pyruvate, so we don't need to eat it from food, I mean, so we can make it from pyruvate, and this is the exactly what is happening in the glycine alanine cycle. Glucose to the pyruvate, and then by the ALT, by the help of a glutamate, to the alpha ketoglutarate acid, then it will form something which is non-alanine. And again, I said L-alanine is more common than D-alanine. D-alanine is only formed in the wall of the bacteria. So I think that is just a simple information about the alanine that we have to memorize it, and maybe we can in the future we make more information, but this is like the simple and the basic thing that we should know about the alanine. Goodbye.